Let me start by asking you a simple but very important question. Have you ever completed an HPLC analysis, saw all peaks clearly, system suitability passed, but then paused and wondered, did I use the correct reference standard? Should I use the API standard? Or the impurity standard? And am I identifying or quantifying? This decision may look small, but in pharmaceutical analysis, it can directly impact data accuracy, regulatory compliance, and ultimately, patient safety. Today, let's understand how to choose the right reference standard, scientifically and confidently. Reference standards are often called the gold standard of pharmaceutical analysis. And for a good reason. They serve as the benchmark of truth. Every result we generate is compared against them. They help us identify compounds, quantify drug potency, and ensure analytical accuracy. More importantly, regulatory bodies like USB, ICH, and FDA expect proper justification for the standards we use. Choosing the wrong reference standard can lead to serious consequences, such as out-of-specification results, investigation, and even regulatory observations. So, reference standards are not just materials. They are the foundation of reliable analysis. In pharmaceutical analysis, we mainly work with two types of reference standards. The first is the API reference standard. This is the pure active pharmaceutical ingredient. For example, paracetamol reference standard. It represents the drug itself. The second type is impurity reference standard. These are specific unwanted substances, such as degradation products or process impurities. For example, paracetamol impurity A, B, or C. Each serves a different purpose, and selecting the correct one depends on what you want to analyze. Now, before selecting the standard, we must understand the difference between qualitative and quantitative analysis. Qualitative analysis focuses on identification. It answers the question, what is this peak? We compare retention times between sample and reference standard. If they match, identification is confirmed. Quantitative analysis, on the other hand, focuses on measurement. It answers the question, how much is present? Here, we calculate concentration using reference standards. Understanding this difference is essential, because the purpose of analysis determines the standard selection. API reference standards are mainly used for two purposes. First is identification. For example, if your sample shows a peak at 5.2 minutes, you inject the API standard. If it also appears at 5.2 minutes, you confirm it is the API peak. Second is assay determination. This measures how much active drug is present in your sample. This is critical for potency testing. But there is one important caution. Avoid using API standards for impurity quantification unless relative response factor is properly applied, because impurities and APIs do not always respond equally. Impurity standards are essential when measuring impurities accurately. Each impurity has its own detector response. Some impurities may give stronger signals, others weaker. If you use API standard instead, the impurity result may be overestimated or underestimated. This affects data accuracy. Impurity standards are especially important in related substances methods and stability studies. ICH guidelines require accurate impurity profiling, which is only possible when proper impurity standards are used. This ensures product purity and patient safety. Now, what happens if impurity standard is not available? This is a common situation. This is where relative response factor, or RRF, becomes important. RRF corrects the difference in detector response between API and impurity. The impurity result is multiplied by the RRF correction factor. This gives accurate impurity quantification. But remember one important rule. RRF must be scientifically established. It should come from pharmacopoeia, validated methods, or proper experimental data. Never assume or guess RRF. And always document the source. Let me share a real industry scenario. In a stability study, an analyst used API reference standard to quantify impurities. The results looked acceptable. Everything was within specification. But later, impurity standard became available. When reanalyzed using impurity standard, the impurity level was actually out of specification. This created serious concern. It triggered investigation and potential batch recall risk, all because the wrong reference standard was used. This shows how critical this decision really is. Here are some practical tips you can apply in your daily work. Always check pharmacopoeia methods first. USB, EP, or BP often specify which reference standard to use. Whenever impurity standards are available, prioritize using them for impurity quantification. Always ensure reference standards are valid, not expired, properly qualified, and fully documented. Because good analytical practice starts with good reference standards. Let's summarize everything in a simple way. API standards are used for assay and identification. Impurity standards are used for impurity testing, purity, and safety. Remember one key message. Reference standard selection is not a routine step. It is a scientific decision. And the accuracy of your results depends on it. Because, in pharmaceutical analysis, accuracy is not optional. It is mandatory. Thank you.